Welcome, everyone. Thank you for joining us for today's webinar, Basket Trials, Biomarker-Driven Drug Development. It is presented by Shivaani Kummar, a professor of medicine and oncology and of radiology in the Molecular Imaging Program, the director of Phase 1 Clinical Research, and director of the Translational Oncology Program at Stanford University. My name is Judy O'Rourke, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before I begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Kummar. I will now turn the presentation over to her.
Thank you, Judy, for the introduction. As mentioned, I'll be talking about the so-called basket trials, discussing the underlying rationale, design, and challenges in conducting such trials. So as you're aware, there was a great paradigm shift in oncology with the development of the molecularly targeted agents. The rationale for developing these agents is that the target is important for disease initiation or progression. The agent that you're developing modulates that target, and that this modulation is associated with the desired anti-tumor effect in the preclinical models. This whole underlying scientific hypothesis was borne out with the dramatic responses that were seen when BCR-ABL was targeted in patients with CML. The authors concluded that our results demonstrate the potential for the development of anti-cancer drugs based on the specific molecular abnormality present in human cancer. And so this opened up a whole field of trying to identify genomic markers and genomic targets that can be then exploited as potentially therapeutic targets for the treatment of cancer. So if we try to study cancer and look at somatic mutations, over 350 of the approximately 22,000 protein coding genes in the human genome have been reported to show recurrent somatic mutations in cancer. And so if we study these and try to understand which one of them are the true drivers of the cancer, then those could potentially be therapeutic targets. With the technological advances and the dramatic decrease in the cost of sequencing and our ability to perform the sequencing in a clinically relevant time frame, the number of tumor samples that are being sampled and sequenced has grown exponentially. The cost, for instance, for doing the human genome was approximately $3 billion, which was a massive international effort. Currently, you can get a targeted sequencing panel done for a couple of $1,000s. What that has done is there's been an explosion of information about the genomics of cancer. So COSMIC is a catalog of somatic mutations in cancer that was launched in 2004 and detailed information on 4 cancer genes. By 2014, it became one of the largest and most comprehensive resource and had information in over 1 million tumor samples with over 2 million coding point mutations listed in the database. So when you look at all of that information, what that has done in the clinical oncology realm is that we have gone from talking about just pure histology-driven classification of cancer to now adding on a layer of the genomic driver mutations. And this is especially true in the case of lung cancer, where for the non-small cell lung cancer, we used to talk about squamous cell adenocarcinoma and large cell non-small cell lung cancers. But now we are also adding on the layer and talking about EGFR positive, ALK, ROS, and additional genomic markers. This is an example of Vemurafenib when it was developed, which Vemurafenib targets BRAF V600E mutations. It was developed initially for melanoma. And in the initial phase 1 study in the pre-selected enriched patient population that had melanoma that was BRAF V600E mutation positive, very high response rates were seen in that trial. And this drug was rapidly developed and approved for this indication. This also brought home the point that it shifted the paradigm for phase 1 trials from going from an unselected all-comer solid tumor population to a population that actually carried the marker of interest and the concept of patient enrichment. So in the traditional drug development paradigm, we used to talk about developing agents for a given tumor type, a given histologic type, and talked about treatment where a particular new therapy would fit into the treatment paradigm. From there, we shifted to talking about a biomarker subset of a given tumor type, for instance, HER2-positive breast cancer, HER2-positive gastric cancer, or Vemurafenib, like I showed in this example, for the BRAF V600E mutation-positive melanoma. But then the question became that BRAF V600E mutations are not only observed in melanoma, however, they are seen maybe with lower frequency, but in other tumor types. So what is the most efficient and best way to evaluate Vemurafenib in the rest of the tumor types? And one of the trial designs that emerged as the one that was eventually used are the so-called basket trials. What these trials are is that you have a genomic abnormality and drug pair that you want to test in multiple tumor types that carry that particular genetic abnormality. So patients are screened, and if the tumor carries that particular quote-unquote actionable mutation, then the patients are treated with on study. If they don't have that actionable mutation, then they are off study. So Vemurafenib basket trial was designed, and as shown here, the way the patients are screened is they come in, the tumors are sampled, and if they carry the BRAF V600E mutation and have, say, lung cancer, they are enrolled onto the lung cancer cohort. If they have cholangiocarcinoma, they'll go onto the cholangiocarcinoma cohort, and so on. And then there is one cohort that is, quote unquote, a basket for all other solid tumors that carry this particular mutation. What this allows is that each of the cohorts is independently powered, and you can draw a conclusion if Vemurafenib is active for that particular tumor type. Also, because there are so many rare tumor types, the cohort that actually takes all the solid tumors would allow you to enroll patients with rare tumor types and evaluate this therapy in those tumors. As shown here, there's a variation in the level of activity observed, especially in the right lower panel, where there are multiple tumor types. There's some that respond and some don't. Interestingly, one of the rare disease, Erdheim-Chester disease, which has a 50% incidence of having BRAF V600E mutation, there was a high response rate seen in this rare indication. I want to shift now to a target that was recently validated and a drug approved against this indication, which also went through a basket trial to show its activity. And then I'd like to compare the two, the Vemurafenib and this particular trial, to see what were the differences. So tropomyosin receptor kinase signaling pathway is an important pathway through which neurotrophins work that are important for promoting sympathetic nervous system development. And they're important in pain signaling, thermoregulation, regulation of movement, memory, appetite, body weight, and proprioception. So in general, the NTRK receptor gets activated. It has an extracellular ligand binding domain, a transmembrane domain, and a tyrosine kinase domain. So when there is ligand binding, it gets activated and signals downstream. However, if there is a somatic chromosomal rearrangement with a genetic fusion that occurs in the 5' end, there is constitutive activation of NTRK, and that results in oncogenesis. NTRK gene fusions have been found in multiple tumor types, and interestingly, some of the rarer tumor types have a higher incidence of having NTRK gene fusions, such as secretory breast cancer and infantile fibrosarcoma. So in order to evaluate an agent against tumors that carry the NTRK gene fusions, and because there are so many tumor types that actually have these TRK fusions at a lower frequency, roughly around 1 to 2%, the best trial design was felt to be a basket trial. For the agent that was being developed, which is larotrectinib, which is a highly selective TRK inhibitor, it has a high potency against all the 3 known TRKs. And the trial that was instituted was this phase 2 basket trial, where we had independent cohorts for lung cancer, thyroid, sarcoma, colorectal, salivary, biliary, and primary CNS, all tumor types where there's been reports of NTRK gene fusions, and then another cohort that had all other solid tumors carrying this NTRK gene fusion. This is one of my own patients, and as shown here, this patient is a non-small cell lung cancer patient that carried the NTRK1 gene fusion. And as compared to baseline after cycle 4, you can see there's a dramatic reduction in the lung mass as well as the liver mass. This is a case of an infantile patient with an infantile fibrosarcoma, a 2-year-old female who had combination chemotherapy with disease progression and was being considered for leg amputation. However, genomic analysis revealed the NTRK gene fusions, and the decision was made to proceed with larotrectinib treatment. After 4 cycles, there was significant reduction in the tumor mass, and patient was referred for surgery. Surgical resection of the mass revealed a complete pathologic response, and the amputation was averted, and the patient continues to do well. So responses were seen across tumor types, so it did not matter which tumor type, and over 17 different histologies were evaluated. And also interestingly, it did not matter which was the gene fusion partner. There have been multiple gene fusion partners reported for the 3 NTRKs, but it did not influence the response rate. And the response rate has been quite durable in majority of the patients. So this data resulted in the FDA approving larotrectinib for patients with advanced solid tumors carrying the NTRK gene fusion. This was a tissue agnostic indication that was provided by the FDA. So if you consider the basket trials for Vemurafenib and then for larotrectinib that I mentioned, one of the things that sort of comes across is tumor histology important? As I had mentioned in the Vemurafenib data, there was a variation in response rate depending on the underlying tumor histology, even though all patients did carry the BRAF mutation of interest. However, in the larotrectinib data, that was not the case. So whenever we're thinking of designing a basket trial or developing an agent against a genomic driver, we do need to consider the preclinical data that supports evaluation of various histologies. But I think based that probably the importance of the target is still disease context dependent. And I think it's important to initially evaluate it as part of a basket trial where there are independent cohorts based on histology. And if it turns out, like in the larotrectinib data, that the response rate is not affected by underlying histology, then data can be pooled together to derive the clinical outcome. One of the other things that other ways to evaluate are what are called the so-called umbrella trials, and some people call it the multi-drug basket trials. But what these are is that patients have multiple different tumors, different histologies, and they have different mutations that are detected, and you have a panel of drugs. So if you say if the patient has a mutation X, you pair it up to drug X, and a mutation Y, you pair it up to drug Y. And patients are treated on study by these multiple mutation drug pairs. If patient has no actionable mutation, then they are off study. Multiple umbrella trials have been conducted and some are ongoing. These are very large efforts with hundreds of patients paired to multiple treatments. One of the studies that was an international effort called the SHIVA Trial, initial data was reported for 741 patients, randomized to targeted treatment versus physician choice. In this, the targeted treatment was a hormone receptor modulator, the PI3. We were trying to target the PI3-AKT-mTOR pathway and the RAS-RAF-MEK pathway. There were 11 different targeted treatments evaluated and data reported for the initial 195 patients that were randomly assigned. 99 were assigned to the experimental group and 92 to the control group. The median progression-free survival was 2.3 months in the experimental group and 2 months in the control group. So there was really no difference based on all the selection. This raised a lot of questions about, is it worth making all this effort for genomic selection of targeted treatment? In cases where we know that something is a true driver, mutation or fusion, then I think it's very clear that doing genomic sequencing and treating that patient with a targeted agent against that driver will result in clinical benefit, as shown with Vemurafenib and larotrectinib. The only caveat, as I mentioned before, is that not all drivers are equal. There may be a histology, tissue-dependent context to it. However, the question about the rest of the ones that are unclear whether they're either driver or passenger mutations, the jury's still out whether targeting it would result in clinical benefit. So I wanted to go through some of the considerations as we think about designing these molecularly profile-driven trials and treatment selection. The first is like, how do we define actionability? What do we call a quote-unquote driver or something that we want to target with a targeted agent? So usually actionability is defined as a genetic aberration or mutation if it is oncogenic and or differentially expressed tumor cells, and there is an agent or drug that putatively works against it. So you should be able to detect that mutation or aberration using the panel that you're using for sequencing, and then have an agent that you think hits either that target directly or somewhere downstream of it. So like I mentioned, we need to be clear on what we are calling a driver. Just having a mutation be present does not mean that it is truly driving the tumor growth. So we need to make sure that there is a functional consequence of the mutation that we are detecting. Also, most of the patients, we are trying to get samples, and we do interventional radiology guided tumor biopsies, which have small tumor biopsy content, like shown here. You have areas where there's tumor and then areas where there is normal tissue and supporting stroma. So what should be the tumor content of the biopsy that needs to be analyzed to reach a conclusion of having an actionable mutation from the sequencing? Also, there is this whole data that has emerged about tumor heterogeneity. So there's ongoing regional clonal evolution. There's difference between regions within a given tumor, between the primary and the metastases. And it has also been shown by Gerlinger et al. That the number of biopsies that you analyze from a given tumor, there is an increase in the number of quote-unquote actionable mutations that you detect from that sample. So what we need to really understand is that what are we considering as a representative sample that then needs to be sequenced, and then that data used to drive the eligibility as well as the treatment for the patient. And should we be now thinking about circulating cell-free DNA as another way where maybe multiple cells are shed, so we may have a better handle on tumor heterogeneity than the core biopsies that are currently performed? Also, then the question is, how many of those cells that we are analyzing need to carry a mutation of interest before you'll be excited enough to say that there is an actionable component in the tumor? Some people state that majority of the cells need to carry a given mutation of interest before it's worth treating that patient with a targeted agent and they're deriving some benefit. Others feel that if it is a fraction, but maybe it is the quote-unquote stem cell fraction, then targeting that would be sufficient. What about single versus multiple aberrations? So it depends on what kind of panel you're using and what you're looking for, but very easily you can imagine that you will find multiple genetic abnormalities in a refractory solid tumor. This is data from MD Anderson that showed that the median time to treatment failure was better with a targeted agent if there was one aberration present. However, there was no difference in the time to treatment failure or the response rate if more than one aberration was present between a matched targeted therapy or without matching. This is interesting data, but also it highlights the point that sometimes you find more than one because you're looking for more than one. Also, it depends on what match therapy you're using, and then whether the one is the true driver or not. So I think we need to be careful in analyzing data such as this, because we do need to find out what the functional consequence of each of the aberrations is before we say the presence or absence of an aberration makes a difference. Also, if we find more than one aberration that is actionable by our definition, which one should we target first to derive clinical benefit, and if there is any data for the safety of a combination regimen? I think what is important is for us to understand before we design these basket trials is what is the level of evidence for defining actionability. I showed you example for Vemurafenib and then larotrectinib. Those are agents that directly target the genetic abnormality that we believe are the drivers. There can be ones where you are not targeting that directly, but you're targeting downstream of it. There are others where there may be compensatory pathways that we are as yet unaware of. And then all of this is as good as the drug of interest that you are studying. So I think as we move forward, there is a lot of excitement in the field with genomics, and there have been major advances in the field and in clinical benefit to our patients. However, as we develop more therapies, we need to be aware of all these considerations. There needs to be a lot more work on the functional consequences of various genetic abnormalities that are being described. And then altering that, how does that change the tumor, and how does it result in anti-tumor activity, as well as what is the normal tissue vulnerability or of us targeting these abnormalities. So I think it's an exciting time to be in oncology, and we are developing a lot more promising agents, and genomics is driving a large part of it. Thank you.
Thank you, Dr. Kummar, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 27th, 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.